Next speaker is Imran Haque, Senior Vice President, Artificial Intelligence and Digital Sciences at Recursion. Dr. Haque leads an interdisciplinary group spanning AI, machine learning, computational biology, and computational chemistry to build and apply Recursion's maps of biology for drug discovery. Prior to Recursion, he was the first chief scientific officer at Phenome, leading R&D in developing early detection tests for colorectal cancer and conducted guideline-changing research as the vice president scientific affairs at Council. He earned his PhD in computer science from Stanford University, where he was mentored by Vijay Pandey and Daphne Koller, and his BS is in electrical engineering and computer science from UC Berkeley. And how wonderful it is that the mentor and student are now giving talks back to back. So welcome, Imran. The title of the talk is Decoding Biology with Data Abundance.
Thank you, Samaya, and yes, it really is great to be speaking right adjacent to Daphne. In another homage to the mission of the company, Recursion's mission is decoding biology to radically transform lives, and I'd like to tell you a little bit about how we decode biology. It's funny, like through the course of 2023, the bottom left of that slide has gotten longer and longer as social media has progressively broken down. And I look forward to 2024 where the entire slide is nothing but social media handles. A little bit of background on Recursion, if you're not familiar with us, we are based in Salt Lake City. And we think like a leader in tech bio. We have 5 programs in the clinic right now, 4 in rare disease, 1 in oncology, and a discovery pipeline that's primarily focused on oncology today, and a number of partnerships in the space as well. So in particular, we're partnered with Bayer in the area of fibrosis, and with Roche and Genentech in the area of neuroscience and a single oncology indication, where we actually just had a program optioned by them in oncology a few weeks ago. In particular, in the area of neuroscience, this is one of the largest deals, I believe, in biotech, period, and I think the largest one in tech bio. And like in Citro, we're also working on iPS neuron work here, and we think, based on public data, we are now actually the largest generator of iPS neurons in the world. Beyond the biology side, though, we're also deeply committed to computation, and of course, that's what we're going to hear about today. And we're also partnered with NVIDIA in the area of advanced computation. There are an awful lot of things that Recursion does. Our aim is to industrialize the entire process of drug discovery, not just target identification, not just hit identification, hit optimization, not just translation, but really to bring all of those together in a set of what we call industrialized workflows, where we generate large amounts of data in-house, in the wet lab and in the dry lab, in order to build what we call our maps of biology and chemistry, which I'll be walking through today. We pair that with advanced computation for target binding and target deconvolution, what we call phenomics, our cellular profile technology, as well as assays like RNA-seq, like measuring chemical tractability and DMPK properties, and then stitch that together with automated workflows, where literally the computers and the robots are the ones taking the first rounds through SAR, through the MTA and ordering, in order to optimize molecules before we have humans address the really hard problems later on. And the goal here is to do this at scale. Now, I don't have enough time during this talk to talk about all of these pieces, and so what I'm going to focus on are a couple of bits of this. The way that we generate this data and the way that we build this scaled phenomics platform in order to build these maps of biology and how we use those to transform discovery. So let's take a step back for a moment and think about machine learning as a problem, and particularly machine learning in this space. Why is it difficult? A very generic version of the machine learning problem statement is that given input pairs x and y, we would like to identify a function x such that f of x is usually close to y. And there's lots of tricks in machine learning, many of which you'll read about in the literature, right? But they usually fall into a few forms. One is you find a new family of F, a new family of functions that's predictive, right? And that could be anything from a simple linear model to a decision tree to a universal deep neural network approximator, right? It could be finding a new optimization algorithm over F, right? A better way to find an F that actually fits your data, or to get better forms of X, right? This would classically be called feature engineering, right? Is there better data that you can use to fit a function to your model? But there's one trick that doesn't usually get a lot of play in the machine learning literature, and that's because it's so obvious, and that is just get more pairs of X and Y. And fundamentally, we think that that's actually the problem that you need to solve. The reason that machine learning is hard in biology and chemistry is because we are operating usually in a very data-poor space. If you imagine a complete matrix of every possible target on the columns and every possible molecule on the rows, that data matrix is extremely sparse, even at the largest of large pharmas, because you're only going to run your high-throughput screening deck against a handful of particular targets, and then you're only going to expand particular islands of chemical space around those hits against particular targets. So you end up with a highly, highly sparse data matrix. And I think that the sparsity of that matrix is actually the big challenge for doing machine learning in this space. So I'd like to talk to you a little bit about how we are addressing that and how we think that there are solutions to do this. In particular, what would happen if we just took cells, knocked out each gene in the genome individually, or individually dosed hundreds of thousands, now over 1 million small molecules in multiple concentrations, and then took pictures of those. What you would get downstream of a lot of work is what we call our maps of biology, and I'd like to take you through a tour of how we use those. So let's imagine that you're a biologist at Recursion and you're interested in working on breast cancer. You could take breast cancer, you could look this up in the human phenotype ontology and get a list of genes that are all associated with breast cancer. You see those as the nodes around the edge over there, right? And you'll see a bunch of familiar faces in that list if you're familiar with cancer genetics. Then what you would do is you would say, okay, well, let me put all those genes and plug them into Recursion's data sets, into Recursion's maps. What that will tell us is which genes, or which knockouts of which genes, are phenotypically similar, phenotypically associated with each other. Those are the edges between the green nodes on the circle. Now, what's critical to note here is that those edges are inferred from a machine learning model that doesn't know anything about the identity of the actual gene. So this is something that is purely data-informed, purely data-driven. And once we do that, and we look at the clustering of these edges, you'll see a number of things immediately pop out. For example, right there above the breast cancer label, you'll see a tight cluster of the BRCA-associated genes associated with DNA repair, BRCA1, BRCA2, PALB2, RAD51, and so on. Right above those, you'll see the cluster of RAS-RAF and proliferative control genes, right? So RAS-RAF, PI3 kinase, HER2, and so on. And again, like these are things that you would look at them and you say, yeah, I expect these to be associated. But the wonderful thing is that they pop out completely from the data. The models that are learning this information from the images have no information about the identity of the perturbation applied to the cells or applied to each well. This comes out naturally. And so what we've been able to do is recapitulate literally decades of discovery in genetics in cancer and genetics in proliferation and damage response from a single experiment de novo. But if all we could do was just recapitulate genes or recapitulate targets, even novel target associations, if you see an edge there that you wouldn't expect, that would be nice. But what we'd like to do is discover drugs, not just targets. And we'd like to do so in a phenotypic and a phenotypically informed manner. So let's zoom into one of these clusters, that RAS-RAF cluster at the top. What I'm doing here is pulling out those genes and now putting on some orange nodes. These are small molecules that we have also dosed on the platform, that we've dosed independently of the genes. And once again, the edges are drawn by phenotypic associations between these perturbations. And again, from a model that is independent of knowledge of what the particular perturbations are. What I've put up here are a number of control molecules. Molecules that should be ground truth that you would expect to light up. And so, for example, we see at the bottom encorafenib and dabrafenib showing up with the edges that you would expect against BRAF. We see compounds, the clinical stage compounds like AZD4547 showing up again against FGFR, TGF-beta, and the edges that you would expect from these compounds. So what this should do is give you confidence that you can actually use this approach in order to drive not just genetic target association, but small molecule discovery and small molecule association. But again, if the only thing that we could do was do this on highly optimized compounds, things that are already ground truths, that would be a nice validation experiment, but wouldn't be all that interesting for discovery. So let me fill in the edges in this graph. If you imagine that this was not an area of cancer where people had been working for decades, where we didn't already have these optimized molecules, what I've done now is fill in the rest of the graph with the small molecule starting points, the NCE starting points in our compound library that also show significant associations with these phenotypes. If we hadn't done decades of work in this space, and for example, those labeled compounds didn't exist, Recursion would actually start its programs from these sets of associations and then work to systematically optimize them on the platform. Now, this is of course a very small slice of the data that we have and the data that we collect, focused on really a handful of genes. There are literally not enough pixels on the screen to show you the full breadth of the trillions of relationships that we've built in our maps of biology. But if I could schematically represent it, it would look something like this, where we have associations between human phenotypes and genes and compounds in yellow, where we have associations between genes in green, compounds and genes in red, and soluble factors in large molecules and these other groups in purple. And so the upshot of all of this is a complete change in how we think about the data matrix that can be applied to drug discovery and to do machine learning. Like I said at the beginning of the talk, in traditional target-based discovery, you might screen a high-throughput screening deck of 1 million compounds and do it against a single target. You might even have to engineer your assay for each new target that you're developing. What we do at Recursion is twofold. One, we don't have to re-engineer the assay. We actually use a constant assay for all of the experiments that we run. But in particular, by using a machine learning guided approach, rather than having to screen 1 million compounds against one target, throw away 99% of the data, rinse and repeat for the next target, every time we run a screen, we are able to relate the results of that new compound that we have screened to all of the genes that we've profiled in the past, as well as all the small molecules that we've profiled in the past. And so when we screen 1 million compounds, we don't get the results against a single target, we get the results against the entire genome, and indeed all of the chemistry that we've profiled in the past. For those who are mathematically minded in the audience, I do apologize. That should probably read quadratically, but exponentially sounds better. So at Recursion, that data abundance that we're able to generate enables maps of biology and enables us to turn biology into a search problem, right? Where we can literally just go to an internal app, an internal database, and look up new compound hits, new inferences against particular genes or against particular targets. And I'd like to give you a brief introduction to how we use machine learning to enable that abundance of data. The maps that we build have similarity as their fundamental property. What we want to know is how similar or dissimilar are two biological states. That's obviously a squishy concept, so the way that we think about similarity is in a perturbative context. Two perturbations are similar if they make the cell do the same thing, and opposite if one reverses the effect of the other. I'm really happy that Daphne presented the example on tuberous sclerosis because it fast forwards through a bunch of this. The short version is that there's a complex, a tumor suppressor complex, comprised of two proteins in physical co-association, tuber and hermardin, that collectively act as a negative regulator of mTOR signaling. What that means in a perturbative context is that if you lose TSC1 or TSC2, your effects should look similar to each other because either way you've broken that complex. And similarly, it should look opposite to the loss of mTOR because this is a negative regulator. You've taken off the brakes on mTOR, so that'll look opposite to breaking mTOR. And in fact, that's exactly what we see in our maps. The knockouts of TSC1 and TSC2, which respectively code for hermardin and tuberin, look very similar to each other. They look opposite, blue, to the knockout of mTOR. I mentioned that one of the key challenges here is really an engineering challenge. How do you make sure that you're not rebuilding your assay for every individual target, for every individual set of perturbations that you want to do? Now, there are a number of high content or high throughput modalities that people use today. There are high content imaging platforms, but even there, in many cases, people are changing stains, changing markers from run to run. There are also platforms like RNA-seq, bulk or single cell, and proteomics. We choose to focus on cellular morphology. And the reason for that is that cellular shape, cellular function are downstream of RNA and protein activity, and we think that in many cases, they can observe effects that molecular assays would not. I think this is something that is going to be familiar to many people here. Anne Carpenter of the Broad, of course, pioneered this in her cell painting approach and is a member of our scientific advisory board. Another advantage is that these images can be super, super cheap. We're talking about an order of magnitude or more than competing molecular approaches like RNA sequencing. And to address a re-engineering problem, we use a standardized assay related to but distinct from cell painting that we call phenomics, where we stain 6 common cellular substructures. That assay is held essentially constant over our different platforms, which enables us to relate the data that we generate to each other in different screening campaigns in different programs. The challenge, of course, in working with image data as opposed to things like RNA data or proteomic data is that images are naturally unstructured, right? It's not like we're getting a tabular view of which genes are expressed at what levels. We have a bucket of pixels. And so a trick that will likely be familiar to people, we use machine learning models to derive embeddings that turn these unstructured images into computable data. In particular, we're able to use these deep learning algorithms to extract embeddings that obey 2 particular constraints. One, they're biologically meaningful. Biologically meaningful here means that, for example, similar perturbations cluster similarly to each other, that we can see similarity in that perturbative context like I mentioned before, but also that they're quantitative, that we can actually see continuity in dose response, and that they're sensitive so that we can do things like compound optimization. Additionally, with the scale of data that we're able to generate and the size of the models we can make, these models can automatically correct a number of challenging issues like batch effect that typically show up in high throughput data. We've shown the power to accelerate development, not just within single cell types, but indeed across cell types. And these get better with more data, and I'll talk more about that later in the talk. So let's take an example of how we actually apply this to do discovery in one of our programs beyond the sketch that I showed at the beginning. This is a program, Target Gamma, which we disclosed as RBM39 earlier in the year, a program in our internal pipeline for oncology. So the background here is that there's a particular gene, CDK12, a cyclin-dependent kinase, that has been advanced as an interesting target to improve response in homologous recombination proficient cancers, particularly HR proficient ovarian cancers. The challenge here actually is intrinsically chemical. CDK12 inhibition, or selective CDK12 inhibition, is extremely challenging to achieve, and in fact, most of the time that people have tried to do this, you end up finding a molecule that inhibits not just CDK12, but also CDK13. CDK13 inhibition leads to a great deal of off-target toxicity that you'd like to avoid. So what we decided to do was ask, well, is there another way to go after CDK12? What we did was looked up CDK12 in our phenomaps, in our maps of biology. And you get a figure that you see here at the right. The first thing that I'd like to highlight is that you'll see in the bottom right corner that there is a very low phenotypic similarity between the knockout of CDK12 and the knockout of CDK13. What that means is that we really do believe that the off-target toxicity that people have observed with dual CDK12, CDK13 inhibitors is probably not an on-target consequence of CDK12 inhibition. There's really reason to believe that this is coming from loss of CDK13. And so if we could achieve selectivity, that would be potentially an interesting option for patients. The other thing that we noticed was that knockout of another gene, RBM39, shown in the second row over there, looked extremely similar to the loss of CDK12, but if you pan your eye over to the left, looked completely dissimilar to the loss of CDK13, suggesting that there was a backdoor pathway to go after CDK12 inhibition by instead targeting RBM39. The third interesting thing that we observed was that a starting point in our chemical library, Rec 65029, showed dose-responsive similarity to RBM39 and to CDK12, and thus represented an entry point for us to actually work in this space. Rec 65029 also showed essentially no similarity to CDK13. And so this story really encapsulates how you're able to use these genome-scale maps of biology not only to do target expansion in challenging spaces, but also to initiate and advance small molecule discovery in these areas. So let's come back to the machine learning. I showed you now how data abundance enables maps of biology. When you generate enough data, you can train machine learning models that extract interesting embeddings from these and then feed those back to build these relational maps that power drug discovery. It turns out that when you do that and you continue generating the data, that further enables machine learning in a virtuous cycle. So for folks who are interested in doing work in the space, I encourage you to check out a data set called RxRx3. We released it at the beginning of the year. We believe it is the largest data set of its kind. I think in its entirety, if you were to download all of the images, it would be about 100 terabytes in size. It has CRISPR knockouts of most of the genome, as well as dose response images from 1,600 approved or commercially available bioactive compounds. We've also released an application, MolRec, that enables folks to explore compound and gene relationships in this RxRx3 dataset. We've released the images, we've released metadata. There are also deep learning embeddings for people who would like to work with something a little bit more tractable than 100 terabytes of images. You can find those all at rxrx.ai. Now, that's a very, very large data set, 2.2 million wells of experiment, and yet that is about what Recursion is able to generate in a single week of running the platform, and it's 1% or less of the total amount of data that we have generated. So we asked the question, what if you could take RxRx3 and in fact much larger data sets we've generated and then use those to make machine learning even better? Well, what we did was use that to build what we think is the world's largest phenomic foundation model. The model exploration here was enabled by BioHive-1. This is, as far as we know, the fastest supercomputer that is wholly owned in biopharma. 450 GPUs, mostly NVIDIA A100s, number 125 on the worldwide top 500 list, on the most recent top 500 list. So we said, okay, what if we take a huge amount of data, a huge amount of compute, and see what we're able to do? What we did was build also a self-supervised model. This in particular was a masked autoencoder built as a vision transformer. And when I say masked autoencoder, what I mean is it's a model that's trained to solve this particular problem. On the right column, we see the original images, crops coming from the cellular images that we take. On the left is the input that's given to the model. And the task that the model is given is try to reconstruct the original image on the right from that masked image on the left. You can see that when we've masked 75, 90% of the image, by the eye, there's not a whole lot there. And it turns out that we have a couple of generations of models shown in this figure. These models can do extremely well at reconstructing these images. There are details available in the preprint that we show there at the bottom, which has recently been accepted to workshops at NeurIPS. But the short version is this is not overfitting. In fact, the validation losses in many cases actually look even better than the training losses. But the proof is in the pudding. What happens when you actually take the embeddings from these masked autoencoders and then throw them at problems of building biological maps, of recovering known biology? The answer is that when you compare them to the models that we used to use, the models that I showed on the previous slide, which were built by weak supervision on convolutional neural networks, they outperform on being able to recover known relationships on genome-wide biological data sets. And this holds true regardless of the data set that we're looking at, regardless of whether we're looking at Quorum, HuMap, Reactome, StringDB. Those red lines over there showing the MAEs dramatically outperform the blue lines, which are our previous weakly supervised CNNs. The other neat thing is that this scales. You'll see a number of different shades of red on this plot. Those are sequentially increasing model sizes and sequentially increasing data sizes, and it improves the performance of the model. You can see all the details in the paper. In the paper, but the short version is that as we scale the training data set from RxRx1, a data set that we released in 2019, RxRx3 at the beginning of the year, and this 53 million internal data set, we see the metrics consecutively improve as we increase performance on these data sets. We've also benchmarked this against results elsewhere in the literature. The bottom row there is a great paper from In-Citro talking also about self-supervised cellular learning. This is not entirely a fair comparison, a number of differences in the cellular platform there, but we agree that the direction forward is to use self-supervision and benchmark on these large-scale data sets of genomic knockouts to evaluate the performance of these models. The closing thought on this area is a quotation by Rich Sutton that I think a number of you in the room will be familiar with, right? What he said was, the biggest lesson that can be read from 70 years of AI research is that general methods that leverage computation are ultimately the most effective and by a large margin. So rather than spending a lot of time doing detailed feature engineering, really hyper-optimizing the models, get a lot of data, get a lot of compute, and in the long run, that's a strategy that wins. And so this is, again, one of the figures from our paper where we work to evaluate the suitability of this bitter lesson in our test problem. The x-axis is showing the total compute scale applied during training, where generally, as a model gets larger, as the data sets get larger, you move over to the right. On the bottom left, you see the performance, and this is in string DB, but it replicates generally across the data sets, of using publicly available data, RX, RX3, and these smaller models. If you simply scale the model size and throw a bunch of, or sorry, if you solely scale the data size but not the model size, you get what's there in the bottom right. You run out of model capacity. It just doesn't have enough to learn. But as you scale both model size and data size, you see performance going up, and you see that line continuing to go. We've not yet seen this to saturate, and we think that that's really what powers this premise that we have, that the combination of scaled data generation and scaled compute is a key to advancing biological machine learning. There are lots of machine learning tricks. At the end of the day, the best one is still get more XY pairs. So in conclusion, one thing that I would like to highlight, particularly for folks interested in doing research in this space of perturbative maps, whether with RX-3, the JumpCP dataset, other datasets in the space, benchmarking on a consistent and diverse range of biology really is critical. Last year, we wrote a paper with our collaborators at Genentech that went to LMRL at NeurIPS, and we've released both the paper as well as code bases to assist in building these maps, so baseline implementations of these maps, and benchmarking them. And we'd really love to help align the field around a standardized set of benchmarks to figure out how we're going to advance the state of map building. Furthermore, I encourage you to take a look at RX-3 as a data set. And for folks who are interested in collaborating, would love to chat with you, in the breaks to see, both talk to you about this data set and what you'd like to see in future data sets. In conclusion, biology, I think, is today's frontier for machine learning. Standardized imaging is incredibly data-rich and scalable, enables these dense data matrices. It enables a number of downstream applications in drug discovery. And John Masefield said that all he wanted was a tall ship and a star to sail her by. All I want is a tall stack of data and the compute to learn her by. We are hiring, and I'm happy to take any questions. Thank you.
Thank you, Imran. That was a fabulous talk. I agree. More data, more power. Please come to the mic and ask your questions.
Thank you for your amazing talk. So you showed us abundant data sets aiming for 1 million compounds times the genome. So how can we capitalize these data sets for design and optimize combination therapy?
So that is a very interesting area of research that we're working on advancing, right? Both testing individual perturbations, as I mentioned, but also doing combinations of perturbations, whether genetic or chemical. And the goal on the back end of all of this is actually to build large-scale machine learning models to do this prediction. So I focus today on the machine learning work that we're doing in cellular imaging and phenomics. There are also additional libraries of work on things like transcriptomic machine learning models, multimodal models, as well as chemistry, in order to do exactly what you're suggesting, right? In order to be able to predict what are either combinations of small molecules or new designs of small molecules that might better elicit the phenotypes that we're interested in.
Thank you.
All right. There's one question on the chat on the webinar. Does Recursion work with bright field imaging, and also, do you have patient iPSC neuron microglia for neurodegenerative disease discovery expeditions?
Yeah, so the answer to the first question is absolutely yes. And bright field as well as multi-time point imaging are significant directions that we're working on and where we see the future. In general, I think as Daphne very nicely illustrated in her talk, right? H&E was a data modality that people thought, oh, there's not that much information in it. You've just got like these two gross stains. But the power of machine learning to extract signal from that is incredibly powerful. And we think that lesson is replicating, in fact, in fluorescent imaging as well, and really enables bright field as well as multi-time point methods that are very useful for things like learning causality. With respect to the particular cell types that we're using in our collaboration, I can't share all of the details, but there's a wide variety of iPS cell types that we're working with there.
Hey, Imran. This was a really great talk. Thank you. One question I had was, are you looking at doing things to change image quality as opposed to image size? Like, are you looking at different technologies that will give you higher quality data?
Yeah, so quality is an interesting question, right? So we have certainly done a ton of optimization in our labs of microscopy protocols and hardware, and do you look at 10x or 20x or 40x imaging, right? How long is your acquisition time, all of that. I think one thing that is really important, and one of the things that's highlighted really by the mention of that benchmarking work at the end, is that what you care about really is the end-to-end quality of the system. And I think one of the lessons that we have learned is that you can get an awful lot of power out of a system that initially appears to be less informative if you can acquire more data with that objective and feed that into a better software system. So rather than thinking about 40x, 80x super resolution microscopy, right, where looking at the image itself seems like it has a lot of information, seems like it has a lot of content, it turns out that a faster but lower resolution microscopy protocol can actually work better because it enables you to have more powerful software on the back end to make the end-to-end system look better.
Thank you.
Yes.
Hey, Imran. Fascinating talk. Thank you. You focused a lot on your phenomics piece of the platform. It's all about data acquisition from an image component. So you have billions of data points, right? But you had, right below that, you had scale transcriptomics.
Yep.
I want to pose a question to you as, how do you guys use molecular data in transcriptomics, proteomics, that kind of modality to inform your models?
Yeah, so you can think about this in a couple of ways, right? So there's one way that you can think about it in sort of a simultaneous multimodality, right? Where you have both morphology as well as transcript, right? Informing the nature of a phenotype and its relationships to other perturbations that you've looked at. You can also think about sequential multimodality, right? Where you can use things like RNA-seq to connect to patient data, or on the back end to validate particular to validate particular predictions that you may have made from phenomics, right? As a confirmatory method to say, hey, make sure this isn't an artifact of a particular assay method, or downstream to then connect to things like biomarker discovery, patient stratification, right? So we have lines of work in each of those, and the methods sort of intrinsically differ depending on the target application.
Thank you. This is really interesting. So I guess you have a platform that could, using only this phenotypic readout, potentially discover selective inhibitors for, something like CDK12. My question is like, at some point you're going to need to do a target-specific assay, if only to please the FDA. At what point do you start to invest in that?
So that's not necessarily true.
Really?
Right?
Okay.
There's a couple of really interesting stories. Like I think my favorite JMed Chem paper from a year or two ago was a compound that got accelerated review and so on. It was in oncology, and I think like 2/3 of the authors, I forget which firm they were from, but it was either like Lockheed Martin or like one of like the defense contractors, right? And they literally screened explosives, and then one of them killed cancer. And they said, we don't really know how it works, but it seems to work, right? There was a paper just a few weeks ago, right, that showed that actually like phenotypic approaches are the ones that have been like much more productive in terms of discovering approved therapeutics than target-focused ones, right? And abundant literature showing that what people think is the target or the efficacious target for a number of drugs is not actually the one driving the work, right? So at the end of the day, like what you want to know is safety and efficacy of a particular molecule, and we are driving scaled phenotypic discovery. Now, beyond that, like certainly there's work that we can do and in some cases have done to assess particular target binding, particular target engagement, right? That was another layer that I didn't talk about here, but through some acquisitions earlier in the year, we've invested in scaled target deconvolution and target prediction in order to identify what are the potential on and off target binding profiles of not just the molecules that we screen in our profiles, but actually computed across the entire human proteome for the entire 36 billion molecules in enamine real. And so that's actually a computational data layer that comes in even before the cellular phenomic screens that I discussed.
So I have a quick question. That is, have you seen any dependence on the morphological change you are observing in those images on the actual expression level of that particular gene in that cell line? The reason I asked is because when we access like jump data, it's mostly done in U2OS cell line, right? But how do we rationalize that? If it is because just the gene is not expressed or it's real change in morphology?
Yeah, absolutely. So it's, it's funny, right? Because there's this constant debate, how much does RNA-seq correlate with protein expression, for example, right? And the same thing is true here. So on one level, the answer is yes, right? If you have genes that are at extremely low levels of observed RNA-seq expression, typically you will not see significant what we call phenoprints. You will not see a phenotype that is significantly distinct from noise when you measure across multiple orthogonal guides and so on, right? However, that does not mean that there are no phenotypes for low expressed genes, right? There are some genes for which translation rates are high off a few RNAs. There are some genes for which there are just very large effects from very small amounts of protein and so on. But you definitely do see a distributional shift between the two. And in fact, that distributional shift was one of the things that enabled us to discover a big bias in CRISPR-Cas9 based screens, where in a few % of cells you end up truncating a chromosome arm. And one of the signs was, we're getting a lot of phenoprints here for genes that shouldn't be expressed. What's going on? And you can trace that down to these underlying assay biases.
Great, thanks. If no more question, yeah, go ahead.
Well, it's my personal question. Is Recursion thinking about increasing the number of channels that you want to, for example, in the image, like using antibody staining to increase the number of phenotypes in the images?
Yeah, so I mean, we use antibody stains, in certain downstream assays, right? If we're trying to validate particular phenotypes and so on. But I actually see the future going the other way, right? And this relates to the question from the chat earlier around bright field, right? I think what we'll see actually is a reduction in the number of channels that are required, the depth of resolution, and instead see imputation by machine learning models filling that in, because that ends up being a highly effective and much more cost-effective way of scaling these approaches.
Hi, Mahmoud Gandhi. Thank you for a nice talk. My question is, how does these image-based CRISPR screen correlations compare with, for example, the data we generate in the depth map, like on the cell line dependency maps? Like if you look at the pairwise correlation for the gene dependencies in the cell lines.
Yeah, totally. So it's distinct, right? Because in the kind of profiling that we are doing, we are looking at a broad range of cellular phenotypes in a single cell type or a single co-culture at a time, whereas DepMap is measuring a single phenotype, right? Differential viability in a large number of cell lines at a time, right? There are assay differences that admit differences in scalability there, right? It's easy to do a pooled genetic screen on these. It's harder to do a chemical screen on all of those in DepMap. But broadly speaking, so that powers two things. One, it's a lot easier to do chemical discovery in the space. And two, there are a lot of effects that you can see that are not simply cell viability, right? In fact, this is a question that we've explored many times over. Oh, are you just seeing cell death? And the answer is no. We can count the number of cells that are proliferating or dying in our assays, and you really do see much more subtle changes in morphology, and those will usually correlate actually with known things about, for example, if you perturb a bunch of lysosomally oriented genes, you're going to see associations with other genes in the lysosome. So it's not just death or life.
Thank you.
All right, so let's thank Imran one more time.